Yes, good evening to everyone. So, welcome to day 12 session on P cell, not P cell, that is on immunological databases and T cell epitope databases. Okay, so before going to today's session on T cell epitope prediction, can we discuss the previous session contents? So I just start with polling. Okay. So I just launched the first question from the previous topics. What was the first vaccine discovered from reverse vaccinology approach? The choices provided are tetanus toxoid, Bacillus calmette-guerin, measles, mumps, rubella, meningococcal polysaccharide. Which was the first vaccine discovered from reverse vaccinology approach? So I think the polling is visible to everyone. Try to answer for this. We just have 20 seconds left out. Very good. I found 8 votes completed out of 13. Excellent. Try. And actually, all the 8 votes are correct answers. Okay. The answer is meningococcal polysaccharide, which is used to treat bacterial meningitis. Okay. Now let me go ahead with the next question. That is class 1 MHC genes. Oh no, it's not launched. Now I launched. Class 1 MHC genes of antigen presenting cells present peptide antigens to helper T cells, T lymphocytes. And next choice is cytotoxic T lymphocytes. Next is B lymphocytes. The finally dendritic cells. We have just 15 seconds left out. So I think I can freeze. I can end the board. The same 8 votes I got. Very good. Try. Again, the majority has answered the correct answer. Cytotoxic T lymphocytes. Okay. And if you see the class 2 MHC genes of antigen presenting cells, these present peptide antigens to obviously helper T cells. Okay, now let me go ahead with the T cell epitope based prediction. Just for 5 or 10 minutes, I show you the PPT, then I take you to the website and tell you how to practically do that. If you see this T cell epitope, just a recap of whatever we discussed on yesterday, we know that a subtype of T cell, that is CD4 T cell, okay, is going to interact with the MHC2. This you have to remember. And similarly, the CD8 of T cell is going to interact with the MHC1 gene of antigen presenting cell. Okay. And the next thing we have discussed something on what is CD4 T cell. It's nothing but helper T cell. And CD8 T cell is nothing but cytotoxic T cell. And like B cell, even dendritic cell is an antigen presenting cell. So it means that the antigen is going to present on the MHC molecules, okay, on the surface of MHC molecules. Obviously, whatever it is going to express on the APC is going to interact with the T cells. And we have seen this as the MHC1 antigen presenting and processing and presentation pathway. This is for MHC1. Similarly, we also have for MHC2 and structures of MHC1 and MHC2. From this, what we inferred? We inferred that we use a small peptide, okay, of around 9 amino acid peptide, okay, which is going to bind with the MHC1. But whereas for MHC2, whatever peptide you're going to bind will be of lengthy just because of two different domains are present on the surface. Okay, if we have 9 amino acid here, generally in publication, you can see 13 or 15 amino acid is taken and related to MHC genes. So class 1 MHC genes, okay, or of HLA-A. So you know MHC stands for major histocompatibility complex, and corresponding genes we call it as human leukocyte antigen. And if you see this class 1 MHC genes, we have basically three important genes: HLA-A, HLA-B, HLA-C. Similarly, in class 2, we can see DP, DQ, DR. All these genes are present in chromosome 6. This is what we have discussed on yesterday. You can find that these genes are present in different locus in the chromosome 6. And now we discuss something on how the HLA is linked with the treatment of diseases, especially if you see autoimmune diseases. Okay, so HLA B27, that is, if any population has this allele. Okay, for example, Africans have these kind of alleles. They are more prone to get ankylosing spondylitis, reactive arthritis, acute anterior uveitis. Got it? Similarly, if a particular community population is going to have HLA-B47, then they are more prone to 21 hydroxylase deficiency. Similarly, for HLA-DR2, systemic lupus erythematosus, SLE, and we can also see the diabetes mellitus type. They are more, the corresponding population are more prone to get these kind of diabetes mellitus if they have HLA-DR3. So, like this, you can see so many genes responsible for causing the respective autoimmune diseases. Yesterday, we have discussed the most important database we use for predicting epitopes is IEDB. It means that Immune Epitope Database. Other than this, an Indian scientist called Raghava, IIIT, so he has brought a kind of a web server, okay, called Raghava web server, where he has having plenty of tools developed for T helper epitope prediction, cytotoxic T lymphocyte epitope prediction, and he has also has so many tools for B cell epitopes and other tools related to vaccine design. Okay, plenty of tools they have developed. So ProPred is one of the tool developed by Raghava scientist. So if you see, this is majorly used to predict MHC class 1. And similarly, ProPred we also have for MHC class 2. So we have different tools, okay, to predict and if you see this net MHC pan, okay, this is another tool used to predict the peptides, epitopic peptides. Like however we have seen ProPred, even net MHC pan is from Denmark Technological University, I believe. So they brought these kind of surveys where we can able to predict the epitopes for MHC molecules. And this is the link how you can able to find in that link. IEDB. This is what we had discussed on yesterday. That for T cell epitope, we have an exclusive kind of hyperlink. If you go to this, you can able to predict the T cells. But whereas if you go to this, we can predict the B cells. Tomorrow we'll be discussing on this. And we also have plenty of epitope analysis tools in IEDB. Now, let me take you step by step. Okay. The first step, what I'm going to make is to take you directly to the IEDB website. Before that, I'm looking through the Uniprot ID. 1 minute. Yeah. You know, Today, we'll be practicing a demo with a Nipah virus W protein, whatever we already worked. Okay, I told you that in reverse vaccinology, we can come up with a kind of epitope based vaccine. Yeah, participants, am I audible to you? Because here I have a low internet strength. I don't know whether I am connected in online or not.
You're connected, sir. You're audible also.
Thank you, madam. So now let me go ahead. Like I was explaining on W protein, I told you we have so many structural and non-structural proteins. I take you to that even that particular slide. If you see this Nipah virus genome, okay, we have proteins like fusion protein, F, G, N for nucleocapsid, P for phosphoprotein. So these are the types of proteins we can able to see, including M matrix proteins. These are structural proteins. We also have non-structural proteins like polymerases and proteases present in the virus. Okay, if you see this Nipah virus, almost all the proteins they have worked. Only one protein which they didn't work is Nipah virus W. That's what I tried. If you see the structural proteins 1, 2, 3, 4, 5, and 6, 6 proteins are available. If you see the non-structural proteins, we have 3. So we are going to try with the W protein. Okay. When compared to SARS-CoV-2, this is a small virus. I mean, it's going to occupy less space, and even the number of proteins present or less in this Nipah virus. Now I'm going to explain you like how to predict T cell epitope from the W protein. So just going to that, see if you see this Nipah virus W protein, the uniprot ID is mentioned is P0C1C7. Okay, it's also mentioned in the journal paper I have circulated to you. P0C1C7. So first we have to go to the uniprot. So I'm going to the uniprot now. So after going to the uniprot, we have to type that P0C1C7, P0C1C7. This is the uniprot ID of Nipah virus W. Let us check whether yes, it confirms that it is a Nipah virus protein W. Here we have to go to the sequence. Inside the sequence, we can able to find the length is 450 amino acid. Simply click download. After clicking download, we are going to take the entire sequence. Later, go to the IEDB tool. Okay, we can better go to the IEDB tool. You know, actually, in the Raghava website or whatever I showed you in that PowerPoint presentation, if you go to the particular link, whatever I pasted even in the WhatsApp as well as in the Gmeet, it directs you to this link. So each and every tool is you can find a link. You can click and you can try with the any protein sequence. Presently, the sample what I'm going to try is the Nipah virus W. So before going to this, let me try initially with the IEDB tool. Okay, so IEDB. So I'm going to type iedb.org. So I'm going to try inside. After getting inside the tool, okay, go to the MSc Class 1 prediction. Before going to the MSc Class 1 prediction, let me first explain what is there in this IEDB. IEDB stands for Immune Epitope Database. Okay, this is the largest database of repository of epitopes of both B cell and T cell. Okay, you can see this much epitopes are deposited already. We can also see peptidic epitopes around 19 lakhs, and around non-peptide epitopes are also present. If you see what exactly we can find in IEDB, other than T cell epitope prediction tools, B cell epitope prediction tools, and other epitope analysis tools, we can also predict epitopes for any diseases, not only restricted to human, but also with respect to animals. So like domestic animals, so dog, camel. Okay, so like if you know that not just the humans are facing or the dreadful diseases, even the animals are facing these diseases. What is SARS-CoV-2? It is a zoonotic virus. It means that it spread from the animals. So what is Nipah virus? Again, it is a zoonotic virus spread from the animals. So then, obviously, the veterinarians can also use these kind of a database to predict epitopes or like to come up with a vaccine even for SARS-CoV-2, SARS-CoV-1, or any other viruses. Okay, so this is the best tool for the most of the species. If you want to know more about this, you have to simply click find. You can able to see. We also have vaccines for birds, especially what we consume most. That is gallus, chicken, turkey, goose, etc. So whatever consumed by human, okay, for that way, the vaccines, that is peptide-based vaccine information are available. In fishes, we have these two category of fishes, and in mammal, we can find a camel, the carnivores. We can also see in zoo. Okay, for few wild animals also we have. Okay, we also have for wild animals. So for cat, dog, badger, so all these things you can find. So this is something about if you want to predict peptides for these kind of a species, we can select a specific thing. For example, gallus. You click apply, and then now you can find any specific kind of epitopes. Okay, so like this you can try. And similarly, you can also able to find based on particular virus and particular protein in the virus. Okay, so these epitopes can also be predicted directly because 19 lakh epitopes are already deposited here. Okay, so one can try this also with multiple filters, whatever available here. We also have exclusively for T cell receptor and B cell receptor. Okay, we can also try like this. Now, first, let me explain about the T cell epitope prediction through MHC class 1. If you see in this MHC class 1, we have for immunogenicity processing binding elution. We have plenty of tools available inside. Now, simply click accept peptides. So, default you can see a sample sequence like SARS-CoV-2 spike glycoprotein, but we want only the Nipah virus. Just copy and paste the Nipah virus W protein. Okay, so whatever we paste, we either paste it in UniProt ID format or you just paste the amino acid sequence. Now, defaultly, for the MHC1, I told you we have a less amino acid peptide. So it means that 9 amino acids. So default it is present in 9. So no need to change. But if you want to come up with a some large length like 10, you just drag the 10. Okay. Now there is no necessity. So I just keep it as such. And the next important input parameter you have to feed is the MHC allele. MHC allele. What is MHC allele? I told you the humans are distinguished. Each and every community population can also be distinguished through MHC alleles. So if how come then we can get the information of MHC alleles? For this, better initially try with this population coverage. Before running MHC1 prediction, MHC2 prediction, check with the population coverage. If you click the population coverage, it take you to the this kind of page. Here you can see East Asia, Northeast Asia. Check whether if you are focusing on India, like if whatever vaccines you going to synthesize, that is from the epitopic based peptides, if it has to treat the Indian population, that to specifically Kerala population. For example, because the Nipah virus has infected the Keralites, isn't it? So then, if you see the India, it comes under South Asia, isn't it? So better I suggest you to go inside this allele frequency net database. Like if you here, if you want to go up to the India, you can try like that. But if I want to go to the specific community population, go to this allele frequency net database. After getting inside, you have to click populations. Inside the populations, browse populations by geographical regions to get inside a specific location. Okay, now I am searching through a geographical region. So if I am disconnected, either give a missed call, something like that. So I'll be getting alerted. So now, because it takes more internet. Okay, now I just got inside this allele frequencies net database browsed by geographical regions. So we have seen that India comes under South Asia. So can we go to the HLA of South Asia? We have only 69 HLA alleles. No problem. But we want to get a vaccine only for Indian Keralites. So I'm getting inside this. After getting inside that, we have to search for the community population. Because we are predicting MHC1 here, we have to look for either HLA-A, HLA-B, or HLA-C. We have to check for any of these three genes. So if you are going for MHC2, then we have to search for HLA-DP, HLA-DQ, and HLA-DR. Now presently we are getting inside the South Asia. I better close other windows. I click once again. I don't know. Yes, I think we got inside. If you see this population, okay, in this population we can able to see the Kerala, but we have different communities in the Kerala itself. Elava, Nair, Tambur 3. Okay, for example, if I focus on Kerala Nair population, okay, specifically, we can click this Kerala Hindu Nair. We have so many Malayalam speaking, you can find ABC, so many genes. I just going to the Kerala Nair. Here, now I am going to click allele frequencies in the frequency data, which is present in the bottom. Okay, now we can see based on the frequency, okay, which is topping the most according to the allele frequency 0.2440, that is oh no 0.3180, that is C07. C07. So this is most frequently observed, isn't it? So now what I'm going to do here is I have to type that HLA A இல்ல C இல்லையா HLA C07. In 07 we find plenty of alleles 07, 01, 02, 03, 04, like that. Whatever you want to select, you select this. Okay, if I want to go with only two, that is 07 and 02 alone, I go with this. Okay, select this. Now two alleles are selected. Okay, so these alleles are we can able to find in Kerala Noyer community population. So after selecting these two alleles, and already this is defaultly fixed as 9 peptide length, we have to simply, so the database prediction method, what we use is the net MSC pan 4.1. Okay, say this because in journal paper you have to give all these parameters. Now simply click run. If you want to give few more alleles, 20 alleles also can give, and you can predict specific epitope peptide for the MHC1 molecule. I am typing the Nipah virus W protein as an input sequence, and from this protein sequence, a part of that is a 9 amino acid peptides are predicted, which can able to bind, uh, which is showing a significant result. Okay, that we take for further screening. Thousands of peptides are generated if you feed any protein sequence. At present, I have fed around 450 amino acid. So it generates more peptides. But if you go with still larger kind of a protein sequence, for example, spike glycoprotein is more than 1200 amino acids, then you can get few more peptides when compared to Nipah virus W. Now we are also received the results. If you see the results here, okay, and if you want to see in the Excel sheet, you can download this. Around 884 rows are available. Okay, so to download the Excel sheet, we can go to the display or download all rows, either in tabulated separated value file or comma separated value file. Both these sheets are going to open in Excel. So now presently I'm going to use the comma separated value file. So whatever I'm going to predict, okay, I'm presently saving it in the folder what we are using from the beginning. This is for Nipah virus W for MHC1, isn't it? So this is the table. So I'm saving the workbook like this. Now I'm going to open this workbook. If you see this workbook, okay, I just zoom in and show you what exactly, and also do the word graph. See, if you see, the peptides are ranked based on net MSC pan EL score. Okay, 0.11. So it's not exactly ranked according to this, but we can able to sort out which is best for this. We can mm, sort out based on the values present in ascending order. Yeah, 0.16667. This is the maximum value of the net MHC pan score. So if you see this peptide AQPBYHWSI, this is the best peptide which is present in the 360 amino acid position to 368 amino acid position in Nipah virus W protein. Okay, so whatever we predicted for the specific allele, this is the best peptide. If you use other alleles, for example, the Tamil Nadu Chettiar, Tamil Nadu Nada, will be getting a different kind of peptides. So this is what we call it as precision medicine. Okay, so even the vaccines can be designed specifically to the community population. But don't restrict up to the one which is giving a top score in NetMHC. So screen around best top 20, because this is only one tool which is predicting. We can also compare with three or four more tools. This is having one particular correlation, and from that correlation, it has predicted which is the best peptide. Okay, and if you see the other tools, they have different physicochemical properties. They consider different physicochemical properties of peptide, which can bind well with that allele. So you take some 20 top ranking 20 epitopes from this tool and compare with other top ranking 20 epitopes predicted from other tools. And if any of these epitopes match with each other, then you can screen that as the best epitope. Okay, only through comparison with multiple tools, we can able to come through a conclusion. Not simply because in journal papers we can't able to just publish that I just use the ATP MSC1 and I'm going to publish, because already these epitopes are there. Okay, there is no such novelty present. Now, I think we can understand how to predict MHC1 epitopes. That is, only thing is that first you have to paste the input sequence of virus and then fix a peptide length. So select the MHC alleles. For this, better go to the allele frequency database. And after that, finding the alleles, you just select here and simply click run and then download this. Okay, you can also see this in the form of peaks. Okay, Okay, graphical chart is not available. Sequence table, only distance. Okay, so we have already done. Now let me take you to how to predict for MHC2. Like however we predicted for MHC1, how to go with MHC2. Before that, I show you I can go with the MHC2, no problem. If I go to the MHC2, okay, so we have to paste once again the Nipah virus W protein. In this case, I have to fix it as 13. Okay, according to John Hopkins University hospitals, they use the 13 amino acid peptide. So I suggest to use the 13 amino acid peptide. In my journal paper, I have done randomly. Whatever I have done, even for MSC2, I predicted for 9 amino acid. That is not good. Okay, so you have to select minimum of 13 and it can also up to 15. According to the literature studies, we can select up to 13 to 17 amino acid. Okay, now I don't want to have a different ranges. If I want to have a single range like 13, so I selected the 13, but you can able to find in the range like 13 to 15. So once again, click here. No, it's not coming. I just drag and keep it here. Now I predict in only one particular length of peptide. Okay. Now I have to type the MHC allele. So whatever I predicted, whatever I searched for the Hindu Nayar, okay, I can able to get only HLA, that is MHC1 allele alone. You don't have any MHC allele, I mean, MHC2 allele. How the MHC2 allele are identified? They are identified as DP, DQ, DR. We don't have that. We just have ABC. So better come outside, come outside. Now you check for the India, Kerala, Malayalam speaking. You can see this DQ and DER. These alleles are prominently present in this Malayalam speaking population. So click this Malayalam speaking. So then we have to get inside the allele frequencies. So if you see based on the most occurred allele frequency, if you see 0.16, but anything else 0.115. Yes, this is most frequently observed 0.16. This is A11. So this is MSC1. This is not in use. MSC1 again not in use. Check with this again MSC1. No. We have to check only DP, DQ here. Still not available here. Only ABC is there. If not there, come back and we try with something else. We have that. Still I can't find that. Can we try something else? For example, Karnataka Kannada speaking. Now let me take you to the allele frequencies. ABC we have. We also have DQB. If you see here, DQB 020201. Okay, so this is an allele, important allele. Okay, for the Karnataka population. So I'm going to paste this DQB 0201. Okay, that is DQB1. We have to type like HLA. Okay, DQB1 02. Yes, it's available. 0202. Like this we have. So like this, you can select multiple alleles, whatever you want to select, because these two alleles are almost same. So I selected these kind of alleles. You select as many number of alleles we can, and then we have to simply click run. So I'm going to predict for Kannada population. That is MHC2 allele for this specific community. So presently, whatever I'm searching is for MHC2. Meanwhile, I can show you something else. Like we have one tool called toxin threat. This is used to predict toxicity of peptides. We already seen Protox 3. You remember? Protox 3 is used to predict toxicity for chemical compounds, drugs, etc. But for peptides, we have to use an exclusive tool like this. So here, if you want to design peptide, we have a separate thing. But for the predicting toxicity, click this batch summation. In this batch summation, you can paste the peptide sequence in a single letter code in a FASTA format. Initially, whatever in Excel sheet I predicted, okay, I got these three peptides as the best peptides, isn't it? If you want to check which is a toxic, which is not toxic, just paste this. I'm pasting peptide in each and every line. Okay, so I pasted just the three peptides. So if I want to predict toxicity, okay, and I want to check toxicity, how the toxicity is predicted, once again through the physicochemical properties like hydrophobicity, charge, pI, etc. Only through this, through the correlation, we can able to predict the toxicity. Okay, we are going to use the SVM method, support vector machine method in the Swiss database. And if we want to display hydrophobicity, charge, molecular weight, these parameters, whatever default is selected, you can keep it as such, and simply you have to run analysis. Now the toxicity is predicted. All the three peptides are non-toxins. And if you want to, whatever properties we have selected, like hydrophobicity, pathicity, pelicity, charge, molecular weight, everything is predicted. If you want a negative charge kind of a peptide, then you can choose this is the best peptide. Okay. So like this, we can also screen the peptides through toxin pred, which can predict the toxicity, and also it can predict the other physicochemical properties. Okay. So now let me take you to the MHC2. Yes, we got the results for MHC2. To download the results, download all rows CSV. So whatever I'm going to predict is now for MHC2, MIPA virus W MHC2. So I save like this. So whatever I saved, I simply open this. Okay, zoom in. Just going to do the wrapping of the text. Okay, now if you see, the most prominent is the net MHC2 pan score. Okay, if it is not sorted, you can sort. It's already sorted, I believe. Okay, it's already in ascending order. So the base peptide, the 13 amino acid peptide shown for this specific allele, the Kannada speaking population, is this, which is present at the top. Okay, this is a 13 amino acid peptide, 251 to 263 position of the same Nipah virus W. Next to that, you can find this 376 to 388. So simply select these three peptides. And if you want to check even toxicity similar to however we have tried, go back, okay, paste these peptides also. And if you want to screen, so simply click run analysis. So the same parameters. Again, whatever the peptides we predicted or non-toxins. Okay. So like this, we can able to check these parameters, properties. Okay, so now let me take you further. What else is there? Uh, so if you see here, we have completed up to this, isn't it? So we have seen like how to predict MSC class one. I told you simply go to the tool and then do all these steps in this link. The next thing is like population coverage for getting the specific population through allele frequency in a database. And this is the paper, you can use it as a sample to work with it. And we also published a kind of a book chapter later to this, so we can also find this information more on this. So Nipah virus, this is the pipeline actually. Started with the protein W and we identified T cell epitopes, and whatever T cell epitopes we screened and finalized, we ultimately have to talk with what? Human HLA only. Like whatever HLA C molecule of Keralites, okay, whatever gene sequence we can able to find from NCBI Gen Bank, take that protein sequence, model it using Swiss modeler or ITSR. So download the 3D structure and then you try to dock the peptide with the protein through peptide protein docking servers. Okay, this is how we can able to find. So this is what we have done in the research paper. So we already discussed about the types of proteins and T cell epitope prediction, how is it done? So theoretically, if you see, they use features such as ampipathicity motifs, okay, that binds to the MSC molecules. For this, we are using the machine learning techniques like ANN, SVM, etc. And here in the MSC1, we also can, other than MSC1 prediction, we can also identify TAP score. TAP, I mean, transporter associated with the antigen processing. You remember? So after proteasomal degradation, which is happening in the antigen presenting cell, the peptides are passed through this TAP, okay, the transporter. So one even through this, and we can able to measure the antigen processing happening in the antigen presentation cell, APC. Okay, so we have a separate link for that. And for predicting the T cell immunogenicity, this is the hyperlink. So for predicting the T cell immunogenicity, okay, how is it done? What are the most important parameters it considers? It considers the antigen processing similar to TAP, and then peptide binding to MHC molecules, and recognition by a cognate T cell receptor. So these are the properties uh, used to determine the T cell immunogenicity. Immunogenicity score will be from 0 to 1. Next, conservancy analysis. The conservancy is a fraction of the protein sections that the epitope contains and identifies a correspondence. Like whatever proteins, whether evolved from the same thing or not, and how about the epitopes, whether we find a distinguished epitopes or not. The conservancy analysis can be predicted using this hyperlink. Toxicity, I just mentioned you, we can able to predict using toxin thread. So this is developed by Raghava scientist, and it uses these parameters to predict the toxicity. These are the physicochemical properties, whatever highlighted in red, or the physicochemical properties of the peptide. So how to make it? First, select a kind of a viral protein, and then we can try. And what is tabulated in the journal paper is whatever we predicted. Okay, so I just used 9 amino acid peptide for both MHC1 and MHC2, but you should not use like that. Actually, for MHC2, I have taken the best two epitopic peptides. For MHC1, I got this much. Okay, so whatever, how I screened, I screened with two different tools, NetMHC-PAN and NETAS, cytotoxic lymphocytes for MHC1. Similarly, for MHC2, I have used ProPred and IEDB. These are the tools I've used. So you got the scores only here for MHC2. For MHC1, I got like this. Okay, even that kind of a methodology is not right, but when you try, you can try however I mentioned in this course. If you see this, okay, out of these peptides, okay, 1, 2, 3, 4, 5, 6, 7, 8, out of 8 HLA peptides, I mean MHC1 peptides, 8 peptides, I can able to screen this is the best epitopic peptide, what is shown in blue color. How I screened, So this is, if you see, based on NetMSC scores, you can't able to find, isn't it? Because it is in between value, intermediate value. It's not a large value, it's not even small value, but it is an intermediate value. And similarly in HCTL. But I have screened this epitope. How you know? So if you see the next table, you can understand. So all these eight peptides, okay, whatever I screened, I also run immunogenicity and conservancy analysis. So the immunogenicity score is most important. How much immunity it can able to give, how much immune responses it can give. So if you see that, okay, yes, whatever peptide I am showing at the top is showing the more immunogenicity value. Similarly, conservancy is more, and even it is non-toxic. So I predicted this is the best for MSC1. Similarly, out of these two peptides for MSC2, when I tried the immunogenicity, I got for this peptide is the best. Okay, and conservancy, and it's also found to be non-toxic. So finally, what we have done is we try to dock, we have docked it, the peptide, whatever we have screened with the protein. The protein we have taken is this sequence. We have taken the protein sequence from NCBI GenPept. Okay, this is the database we have used. So in the what is this actually NCBI GenPept? It is nothing but a protein database, protein sequence database. In NCBA, select a protein and just select for the search for the sequence, HLA DR sequence. I got this kind of a sequence that I modeled using Swiss modeler. Later, we docked this peptide with the protein. Okay, and similarly, we had tried with this. Okay, whatever this is for MHC2 peptide. Okay, we talked MHC2 with respect to the MHC2 peptide, and this is MHC1 peptide. So HLA-B, it is nothing but MHC1 molecule, isn't it? So that we docked with this peptide, whatever we predicted. And if you see the score, okay, we got something like here as 0.15 kilocalorie per mole. Here it is minus 1.19. So according to the significance of results, whatever I mentioned you for the chemical compounds, that should be minus 6 and below. But for here, there is no restriction. Whatever scores you get finally after talking peptide with the protein, you can show it. Okay, there won't be any problem. Now this is related to the MD simulation done after talking. Whatever talk complexes we got, okay, we just tried with the MD simulation. We run the MD simulation for 200 nanoseconds, and from the 200 nanoseconds, it is we can able to see that this peptide is binding stable, okay, in this particular time, okay. So and after this, you can able to see this. It is stable after it is stable for particular time. Okay, so here the RMSD is around zero point 0.6 something like that, but here it comes around zero point 0.9 something like that. Okay, so lesser the RMSD, more stable. That less time it is more stable, and if we crossing after 150 nanosecond, somewhat less stable. But still, you can tell just because it's less than one, it is more stable. Okay, and now let me take you to the RMSF, root mean square fluctuation. Here the fluctuation I got only in this particular amino acids. Okay, this alone you can find somewhat unstable, but it's not perfectly unstable. You know, if and if RMSF is exceeding one, then only you can tell this particular region is highly unstable. So it means that the peptide is binding stably with the protein. That's what we infer. Okay, this we tried with the MHC2 molecule. MHC2 molecule. So whatever we have done, okay, it's just tabulated. Similarly, area. So what is the average area we got? And similarly, how about the radius of variation? Okay, so how much it is stable, the moment of inertia is stable. This is what is tabulated here. That's it. Then I think we have discussed all these things. Now let me take you to the next thing. That is adjuvants in COVID-19 vaccines. Till now, whatever we discussed is related to peptide-based epitopes. How to predict peptide-based epitopes. Now, if you discuss something about the next area called adjuvants, okay, the mRNA spike glycoprotein. So that kind of vaccine was discovered by Pfizer combined with BioNTech, isn't it? They have used a polyethylene glycol, and we can see 95% efficacy with the Pfizer vaccine. Not everyone we receive this Pfizer. And if you see the next coronavirus vaccine, A is at 1,222. You know, when the world is declared pandemic in 2020, that is March 2020, okay, the scientists in the world have taken around 1.5 years to come up with around 89 different types of vaccines. Out of 89 vaccines, the government of India has supplied us two types of vaccines, isn't it? The two types of vaccines, Covishield and Covaxin. But there are also few more vaccines available in the world. So we are discussing like what adjuvants we are using in that type of vaccines. The AstraZeneca and Oxford come up with these kind of a vaccine. For this, they have used adenovirus as the adjuvant. Okay, that itself can be used. The vector can be used as an adjuvant. And in Covishield, okay, again we are using the adenovirus from Janssen's and Serum Institute of India product. And Covovax. For this, we use matrix M adjuvant. What is matrix M? It is a 40 nanometer nanoparticles composed of saponins. What are saponins? These are plant-based secondary metabolites. Okay, this is a major adjuvant used along with cholesterol and phospholipids. Okay, so even our company is selling these kind of secondary metabolites, saponin. And Sputnik, uh, the Russian vaccine, is using an adenoviral vector as an adjuvant, and Sinovac inactivated viral vaccines. So these are adjuvants. Functional diversity in the adjuvants. Past 100 years, more than 120 years, the vaccines, they used the adjuvant like alum, the popular used adjuvant. This is an actually inorganic salt. Okay, the inorganic compound can also accumulate in our blood, in our tissues, isn't it? So, for biodegradable in nature, so at present we are searching for the natural source, that is adjuvants derived from natural sources like plants, animals, and algae, etc. Okay, so our company is also striving to get so many kind of secondary metabolites which can be used as an adjuvants for the, which is most important component for the vaccine. So this is something about functional diversity in the adjuvants. Okay, at present more of plant-based adjuvants are used. Now something related to SARS-CoV-2. In SARS-CoV-2, in second wave mutations, we can able to see out of 1,210 amino acids in the spike glycoprotein, this specific mutation, like in the 417th amino acid, instead of lysine, if it is mutated to asparagine or 309, it can create one different strain. So K417 is one strain, K417T is another strain. Similarly, L452 is another strain. So for each and every strains, we can also come up with the MSC1 peptide and MSC2 peptide for specific community population. If you try like this is actually what a precision-based medicine. Okay, we can design vaccines specifically for Indian community. Okay, even whatever I projected in Nipah virus, with T cell epitope prediction, I also tried with B cell epitope. But for journal paper, the page word limit is there. So I just published the T cell epitope. If you see in Nipah virus, the best epitope is nothing but B cell epitope. For Nipah virus, B cell epitope is best. But that are already published. So I tried with the T cell epitope for publication. And if you see SARS-CoV-2 virus, for SARS-CoV-2, which epitope is best, when you compare T cell and B cell, okay, the best is the T cell epitope. So obviously, it varies with respect to type of virus, whether it's a single standard, double standard, whether it's a negative standard or a positive standard. So all these things, okay, DNA virus, RNA virus, all these things, they matters on what epitope is important for that specific type of virus. Okay, we have plenty of strains in the SARS-CoV-2, alpha, beta, gamma, delta. For alpha, we have around six strains, and beta we have around eight strains, eight mutations. Like this, you can say hundreds of mutations. So obviously, if a patient is diagnosed with a kind of a sequence, like what kind of mutation and what kind of a sequence, then obviously we can also design vaccine for them. Whatever a bioinformatician can design a vaccine, obviously if a peptide based labs can synthesize the peptides, these can be used for prevention for the other the same community population. Okay, so this is how the precision based vaccines are designed. That's it. I've completed today's session, and so more about the other tools. Okay, if you see the T cell helper epitopes, you can see ProBred. Okay, if you want to predict from any of this, simply click here. Okay, paste the sequence like Nipah virus W protein, whatever you taken from the UniProt, and then you try the same. Okay, so this is how we have to try with other tools also. Okay, so you can give the name of whatever you can, and then simply select the alleles. The alleles are present here. You can select the specific alleles and then simply click submit. You can also get in graphical format. Presently, three alleles are selected, and if you want to submit, submit sequence. But I want in graphical format. I rushed. Come back. Graphical submit sequence. So this is a graphical format. Whatever is at the top, this is the best peptide. Okay, which is crossing that red dotted line threshold. That is the best peptide. And if you see, the best peptide is this one, which is present at the top. Okay, like graphical, we can also see the table format. However, you have seen. Like this, we can predict T helper, cytotoxic T lymphocytes. Different properties are mentioned. See, for cytotoxic, prediction of promiscuous binders, identification of proteasomal cleavage, prediction of tap binding peptides. I told you, no, transported associated proteins. So for this, you have to click here and you can paste it. For all these tools, even in cytotoxic lymphocytes, you won't get the same peptide, which is present at the top. Somewhere present in the 35th position in ProPred, that will be in the top one in the tab grid. So I suggest you to go with the top 20 or 30 or even 40, and then you have to use the machine learning techniques. We can use the machine learning techniques to screen based on number of tools you have used for prediction of base peptide. Got it? So here you can use much of machine learning and linear and conformational B cell epitopes and other kind of epitopes, other kind of thing we will be discussing on tomorrow. So today I've completed T cell epitope prediction. If you have any doubts, you can ask me. Participants, thank you very much for your patience. Special thanks just because even on holiday I've joined the session and made it more interactive. So have a nice weekend to everyone. Okay, so we have the next class. We have just two classes left out. Okay, that is one on B cell epitope based databases, how to predict the B cell epitope based vaccine. The other is related to case studies for vaccine design, like how we can even use the machine learning techniques and what are the other tools and servers available for computational vaccine design. So we'll be discussing on the 28th, that is on Tuesday. So we have around 4 days, okay, 29, 30, 31, and 1st. So 1st of August. So within these 4 days, you can complete 25 MCQs. Whatever I asked, 25 MCQs are very basic. So it is feasible to complete it. Completion of overall feedback and 25 MCQs are mandatory to secure the certificate. Any queries? If no queries, thank you. Good night. Have a nice weekend.